So let's move on then to adaptive immunity. We talked about innate immunity, or what sometimes is called like generalized or nonspecific immunity. And now we're going to talk about adaptive immunity. And the word adaptive really just means it's acquired over time as your body adapts to its environment. You begin to develop a more specific immunity directed against very specific antigens that you may have come in contact with. So we'll talk about this side of the immune system kind of as the second group of immunity. So as I mentioned, adaptive immunity is a more specific response, whereas innate was pretty much general, meaning regardless of the antigen that was reacted against, the response would be the same. So we talked about things like inflammation, for example, not very specific to any particular type of injury or infection, but pretty much the same type of inflammatory response to any injury or infection. Adaptive immunity is going to be a lot more specific. It's going to be directed against a particular antigen, and that particular antigen is going to be on a particular organism or a particular molecule, and so we're going to start getting very specific responses. Antigens, when we use that term, most often what we're talking about are proteins that are expressed on the surface of a cell membrane. And that can be an antigen that's on the surface of a bacteria. It can be on the surface of an infected cell that's been infected with a virus, and some of the viral proteins are being transported to the surface of the cell, and they can be noticed or recognized by other immune cells there on the surface. An antigen could even be, we talked about this in the blood lecture, could be not a protein, but maybe a glycoprotein or even just a sugar element, a carbohydrate, like the ABO grouping that we talked about in red blood cells. Those are not actually proteins, but they still function as antigens. So it's typically a protein, but some kind of molecule that the immune system will recognize as foreign, as non-self. And that recognition of a foreign molecule will then initiate an immune response. And because it's a particular protein, and all proteins are different in some way, it becomes a very specific, a very directed kind of response that's directed against only that antigen, and therefore against only that bacteria or only that virus or fungus or whatever it is that we're talking about. And when we talk about adaptive immunity, we break it down into two types. You can see here in the picture, there are really two types of adaptive immunity, and they work together. So we're going to talk about them separately, but they're happening really in each immune response that you have. Typically, you're going to have both of these mechanisms occurring. But we'll break them down so we can understand them separately. So if we look at cell-mediated immunity, what that means is exactly what it sounds like, is that the attack on the foreign substance or against the antigen is going to be by cells. And in this case, really for all of adaptive immunity, we're talking about lymphocytes. And we'll talk about a few different lymphocytes on the coming slides. But so there's going to be a lymphocyte, typically a cytotoxic T cell or something like that, that will actually find these abnormal cells with these foreign antigens and will destroy them. So that's cell-mediated. On the other side, I think is what we think of a lot of times, it's what we call antibody-mediated, or another term for that is humoral immunity. And so that's going to be mediated by antibodies, basically, which you can see here. These are all antibodies. And so the antibodies are going to be kind of critical on this side of the immune system to help identify and tag foreign material and present that to other cells that can destroy it. And we'll talk about this in more detail here in the coming slides. But these are the two kind of primary pathways for adaptive immunity: cell-mediated and antibody-mediated. Okay, so I mentioned that we're going to be talking about lymphocytes really in this whole section of the immune system. So in the general or innate immune system, we talked about a lot of the granulocytes, things like basophils, which become mast cells, or macrophages like, phagocytes like neutrophils, eosinophils, and macrophages. So those are all from the myelogenous blood cell line. Here in the more specific adaptive response, we're going to be looking at the lymphocytic line of cells. So we're going to talk about several lymphocytes. And you can look here on the picture. You can read the words later if you want to. But we can see the primary cells we're looking at are T cells and B cells. We already talked about natural killer cells in the innate immunity. They do function a little bit here on the specific immunity as well, but we're really going to focus on T cells and B cells. Now, B cells are easy enough to remember because we're only going to talk about one kind of B cell. Well, I should say two types of B cells. The B cells that become mature and produce antibodies, these are called plasma cells. So B cells become plasma cells and produce antibodies. B cells can also become memory B cells, and we'll see that in the future. So technically there's two types of B cells, those that become plasma cells and those that become memory cells. On the T cell side, we have a few more options, and we'll look at 3 of the 4 of these. We're not really going to look at regulatory T cells, so you can kind of cross that out for now. We will talk about cytotoxic T cells. Remember, cyto means cell, and so toxic means toxic to cells. So these are the lymphocytes that actually attack infected cells and can even attack any cell that's expressing an antigen. So we're going to see that these are really important in viruses, because viruses will infect our own cells, and then they will express some of those viral proteins on the surface of the cell, and the cytotoxic T cell will recognize those foreign antigens and actually destroy those cells. So these are cytotoxic. They actually are cell killers. Helper T cells are super important because they help activate cytotoxic T cells. They help activate B cells so that they can become plasma cells and make antibodies. So these are not the T cells. These are not lymphocytes that are actually going and destroying infected cells, but they're really important for activating the cytotoxic cells and activating the B cells, and we'll talk about them as we go along. Helper T cells, by the way, these are the ones that are deficient in HIV infection. Memory T cells, these become important for the secondary immune response. So once you've been exposed to a particular antigen, like a bacteria or a virus, some of the T cells that have been activated will kind of remain in hibernation, so to speak, and they'll be waiting for the second exposure. So let's say you get chickenpox at age 5, and then you get exposed to it again at age 20. Those memory cells will be waiting, and they can create a much quicker and a much stronger response, and we'll look at that as well. So 2 types of B cells, plasma cells and memory B cells, and 3 types of T cells that we'll look at: cytotoxic, helper T cells, and memory T cells. Okay, so adaptive immunity, we've already mentioned this, really revolves around this idea of antigen exposure. So you have to be exposed to some very particular, very specific, often protein, and it's that protein that's going to generate the immune response. So antigens are typically on the surface of a cell. So if we think of an antigen on a bacteria, for example, it will be some protein most often that is embedded in the cell wall of that bacteria, but because it's on the surface of that cell, it's able to be recognized by immune cells that may come in contact with it. Now, antigens can also be not from bacteria or viruses, but sometimes our body will recognize, for example, a part of a pollen particle will have an antigen, will have some kind of a protein or glycoprotein on its surface that our body sees as foreign, and it will also kind of mount an immune response as if it were a bacteria. So antigens can be really on the surface of anything, but typically in the immune system we're talking about viruses, bacteria, fungi, parasites, things like that. But I think the key to remember is that they're on the surface, and that's how they are seen. That's how they come in contact with the immune system, because they're on the surface. And once they're detected, then they will kind of produce an immune response. So everything really hinges on an antigen coming in contact with some kind of an immune cell or immune protein. And one of the ways that antigens are found or are discovered is by antibodies. So antibodies are also proteins, but antibodies are proteins that are made by our own body. And they're made from B cells that become plasma cells, B lymphocytes that become plasma cells. And antibodies are very specific to only a particular antigen. So they have binding sites that will only bind to a particular antigen, another foreign protein. So antibodies have a Y-like structure, you can see here in the picture. And you'll always see them drawn this way, and they actually are shaped this way. And they have different parts to them, and I'm just going to point out two parts. We're not going to talk about heavy chains and light chains and all of this. I just want to point out two things, really. One is what we call the constant segment, and that's typically the stem of the Y there. And that's the part that for each type of antibody, and on the next slide we're going to see at least 4 different types of antibodies, but for each type of antibody, the constant segment remains the same. So it's what identifies a group of antibodies. So it's not changing with every antibody. It changes between groups of antibodies, but it's the same within a group. And we'll see what the groups are here in a minute. So the constant segment is not the part that's binding to antigens because it's not specific. It's the same among the whole class of antibodies. The part that's actually binding to antigens is what we call the variable segment. And the variable segment is basically on the ends of the Y. So you could see like here and here. And so you can see actually that each antibody really has 2 variable segments. So it has 2 places where it could bind to an antigen. And this is going to be very important when we talk about the function of antibodies and how they work and how they aid in the immune response. Sometimes they're aiding because they're able to grab clumps of antigen on various cells and kind of pull them together and take them out of solution because they have 2 binding sites. So it's really important to understand the 2 binding sites that they have. And those binding sites are called the variable segments. So you can see here is the variable segment on that side, and then here would be that would be the variable segment on this side. Okay, so I mentioned that there were different types or groups of antibodies, and that's the constant segment. So for example, all IgA antibodies would have the same constant segment. That's how you identify that antibody as IgA. So there are more than the 4 that I've listed here, but these are the 4 main ones that you'll be using in the future, I'm sure. So these are the ones I want to highlight. IgA antibodies, these are the ones that are found in secretions. For example, your saliva has IgA antibodies. And so a lot of kind of our first defense is found through IgA antibodies because they're able to bind to bacteria that are trying to enter through the mouth, for example. Also tears, sweat, breast milk. This is why we talked about breastfeeding is important for that newborn to be able to receive some of its mother's antibodies. Primarily, the antibodies we're talking about are IgA antibodies, and those can be helpful for the newborn who has not yet had time to produce any kind of specific immune response. It still has its innate immune system running, but has not really developed its adaptive immune response yet because it hasn't been exposed to very many bacteria or viruses. So it can get some immunity from its mother through the IgA antibodies in the breast milk. IgE, I'll just mention briefly, and really the only association I want you to know is that they're elevated or useful really in allergic reactions, or we could say not useful. But this is when you're going to hear discussion of IgE is when you're talking about some kind of allergic reaction. Often IgE is involved in that allergy. IgM is important because it's typically the first antibody that is formed once your body is exposed to a new antigen. So when your immune cells encounter, let's say, a new bacteria that you've not seen before, and the immune response is triggered, and B cells become activated and they become plasma cells, often the first antibodies that are made are IgM antibodies. And so if you're looking for signs of recent infection, we will look specifically for IgM antibodies because they're only present in the first weeks of infection. If we're looking for a chronic infection, often the IgM antibodies have disappeared after a few weeks. And so they're really useful in helping us distinguish between an infection being acute or an infection being chronic, or just having a history of a past infection but not having a present infection. So if the IgM is absent, it means that this is either a chronic infection or you were exposed years ago to an antigen, but it's no longer present. So the IgM is useful clinically looking for recent infection. IgG, now this is really the main antibody that our body uses. You can see it's about 80% of all the antibodies that are floating around. This is really the workhorse of the humoral or antibody-mediated immune response. It's the antibodies that are going to do the bulk of the work in helping us get rid of infection. But they take a little bit longer to be formed, and so that's why the IgM shows up first and the IgG shows up a little bit later. IgG will be present in small amounts even after you've cleared an infection, and so it can be useful for looking at, have you ever been exposed to that particular antigen. So I'll give you an example. When I went to medical school, everybody has to, so when I was a kid, we didn't have varicella or chickenpox immunizations like my kids have now. And so they wanted to immunize me against chickenpox because I wasn't sure if I had it as a child. And they said, well, before we immunize you, why don't we just check your blood for IgG antibodies against the varicella virus? And it turns out I had antibodies in my system to the varicella virus. Now, that doesn't mean that at that time I was currently infected with chickenpox. It just means that at some point in my life, I had been exposed to the chickenpox virus, and IgG antibodies had been made against that virus, and there were still some of them in my blood, probably waiting for the next time. And so I didn't have to get the immunization because I had already been exposed. So sometimes we use IgG in the blood to indicate a previous exposure to a particular virus or bacteria. For example, when we're diagnosing hepatitis B and C, those kind of chronic ones, we look for IgG antibodies. But during the immune response, they're really the workhorse. They're really the ones that are doing the brunt of the activity. Here's just a picture from your book. You can look at this. I don't think it says anything different than what we talked about, but you can read over these. We didn't really talk about IgD antibodies, but you can look at the other ones here. Okay, so how do these antibodies function? So these antibodies are going to interact with antigens, and make sure you get those two terms separated and clear in your mind. Antigen is the protein or other molecule on the surface of a foreign cell. whereas antibody is a protein made by your immune system that detects and binds to these foreign antigens. So that's what they're doing. They're floating around in your blood, and they're bumping into various cells. And if they bump into a cell that happens to have a foreign antigen on its surface, then they will bind to that antigen. We can see here they can bind to toxins, for example. There are bacteria, some bacteria kind of wreak their havoc by producing and secreting toxins, which are usually proteins that are harmful to your body's cells. And so antibodies can bump into them and can grab a hold of them and can kind of pull them out of the blood so that they don't hurt the cells. Excuse me. They can also bind to viruses. And remember, viruses work by trying to enter host cells. So let's just take a cold virus, for example. It would try to invade respiratory cells, respiratory epithelial cells and such, and would try to use your own cellular, mechanisms to reproduce itself. So the first step for a virus to infect you is to get inside your cells. Well, antibodies can bump into those viruses and detect the foreign antigens and can actually bind to them in such a way that they're unable to then enter your cells and reproduce themselves. So they can be very helpful in preventing viral infections. And we call that process of either grabbing a hold of toxins or binding to viruses and preventing them from infecting cells, we call that neutralization, because they've really stopped the toxin or the virus before it has a chance to do any harm. It neutralizes those antigens. Another function of antibodies is what we might call agglutination, which is really kind of clumping of cells or proteins that contain these foreign antigens. So remember, the antibody has two antigen binding sites, right? One here and one here. So there can be a cell here, for example, that has some foreign antigens, and that edge of the antibody could bind to that cell, and there could be a cell here, and that antibody could then bind to that cell. Now you've got one antibody that's holding together two cells. Now imagine you have another antibody, and it's grabbing a hold of that cell, but it's also caught a third cell. And now I have another antibody that's able to kind of complete that circuit, and I've just now taken 3 bacterial cells, and I've clumped them together, and I can kind of pull them out of the blood and bring them to other immune cells that are able to destroy them. And so we call that clumping. The more clinical term or scientific term would be agglutination. And so this is one of the ways that antibodies function to remove harmful antigens from our body. The third function I wanted to talk about is something called opsonization. And we talked about this already back in the innate immune system lecture, where we talked about complement and how complement would kind of attach or bind to antigens on, let's say, bacteria, and then they would be able to present that bacteria to a macrophage, and the macrophage could bind to the complement, which would help them grab a hold of that bacteria, which otherwise might be too slippery for it to grab a hold of. So antibodies work in the same way. You can see here in the picture that you get antibodies, they've got their binding sites, right? And they're going to bind to these antigens. And when they bind to the antigen, then they're able to, the other end is able to bind to the macrophage. You see here, that could do some binding. And then that allows that macrophage to then kind of surround and form its vesicle so that it can destroy the bacteria. So that's what we call opsonization. So we've kind of talked about the individual parts of the immune response, but I want to kind of put it all together and talk about what's the flow, or how how does the immune response get generated, and how does it move? What's the timeline for adaptive immunity? So this is just kind of an overview slide. And we should really think about T cells as being the first cells that are activated. But in order for a T cell to be activated, it has to be shown a foreign antigen. So the cells that are really kind of out in the tissues, wandering around and bumping into these foreign cells, like bacteria, for example, we talked about those under the innate immunity lecture. So these are like macrophages, for example, or neutrophils. And so they're the ones that are kind of the first line of defense. And so they will consume, let's say we have a macrophage, and let's say he consumes a bacteria, and that bacteria comes in and it gets dissolved, right? And then it kind of gets released by exocytosis. But some of those particles will actually start moving to the surface of the macrophage. And so the macrophage will then present itself to a T cell, and the T cell will recognize that foreign antigen. Now this T cell will then maybe have one of several functions depending on what kind of a T cell it is. If it's a cytotoxic T cell, so if it's cytotoxic, it'll just go and start, destroying the bacteria. If it's a helper T cell, it will go and find cytotoxic T cells and activate them, or it will go and find B cells and activate, them to become plasma cells. So you can kind of see the flow here, and then the plasma cells will begin to make antibodies, which can then bind to the bacteria and either present them to cytotoxic cells or present them to other macrophages. So you can kind of get the flow there of how the adaptive immune response is working overall. So the first part we talked about is antigen presenting, right? So some kind of antigen presenting cell, like a macrophage, that's kind of the poster child for antigen presenting cells. So he's going to present an antigen to a T cell. Okay. Now that T cell, or maybe it was a B cell that bumped into it, but either way, you're going to get a lymphocyte that becomes activated because it has seen that antigen, right? So let's blow up the picture here. You can see. So the picture here is talking about B cells in particular, but let's say there's an antigen and it's sensed by a particular B cell, right? Now there are millions and millions of B cells, and this picture is just trying to give you a sense of that by showing you 6 different B cells. But floating around right now in your body, there's millions of individual B cells that are all different from each other, and they each will bind to only one particular antigen. And so they're just roaming around your body waiting to bump into one of these antigens. So let's say that happens. Let's say the antigen comes in contact with the B cell that happens to, fit, happens to be designed to detect that particular antigen. Then that lymphocyte then responds to the antigen by proliferating, by reproducing. Okay, so we're going to call that, this is kind of the activation here of the B cell, and that results in cell division and multiplication. So it's going to happen in 2 ways, and I mentioned this earlier. One side of this cell division and replication is going to result in memory cells, and we'll talk about them a little bit more in the future, but they're going to help with the next time you see the same antigen. They're going to help you mount a quicker and a more powerful response if you see this antigen a second time. So they're not really going to be helpful in this initial immune response, they're for the next time. Okay, but this other cell line here is going to be useful right now. It's going to be part of the fighting force for this current infection. So you see that you get multiplication and eventually end up with plasma cells. And those plasma cells, of course, produce the antibodies, and those antibodies then go into your bloodstream. But recognize what's happened here. You have selected only one kind of lymphocyte. So you are not making copies of A, B, D, E, F, or the other millions of antibodies that you could be making. You have selected out only one particular lymphocyte that's very specific to the antigen that's currently in your body. And so you're able then to focus your efforts and your energy and your resources on producing only the type of antibody that is needed right now. So these antibodies are specific for that antigen. And this is a really efficient way for your body to run the immune system. So it has a bunch of small, sampling of millions of lymphocytes that are available to be activated. But then it waits for a specific one to be activated before it begins to proliferate and use its energy and resources. I think your book uses this example, but it's kind of like when you go to let's say you go to a bakery, or I've seen this at restaurants, and they want to show you, what their food looks like. So they have one sample of every kind of cupcake or pastry, let's say, and then you say, okay, I want pastry number 3. I want the chocolate croissant. And they'll say, okay, and then they'll go back and they will make however many chocolate croissants that you want, right? Or let's say you're catering for a party. So they'll show you a sample of everything they can make, but they don't make 100 of each of those samples, right? Because they don't need 100 at this moment. They only need to make the one that you want. So when you tell them, I want the chocolate croissants for my party, then they say, okay, how many do you want? And they make 100 croissants for you. So the same thing here, where you have, just a few of each of these lymphocytes swimming around, but once they come in contact with an antigen that's foreign, you can now start producing that particular lymphocyte in large numbers and antibodies in large numbers in order to fight that specific antigen. And to me, this is very efficient. You don't waste energy and resources making millions of copies of each one. You can wait for them to be activated. Okay, so we talked about antigen presentation, right? So it may be for like an antibody, it may just bump into an antigen, but often for T cells, they have to be presented. A cell has to come to the T cell and present it. And this is often happening in the lymph nodes, by the way. There are a lot of T cells and B cells that are just waiting in the lymph node to be presented with antigens. And so lymph nodes tend to drain the area closest to them. So for example, the lymph nodes in your armpit drain the chest wall on that side. The lymph nodes behind your ear and in your lower neck, they drain, the forehead, the ear, the throat. So they're picking up, they're going to be kind of part of that first response to local infection. So the macrophages or neutrophils or whatever that are encountering these bacteria, let's say, in the local tissue, will then go to the nearest lymph node and present these antigens to T cells. So then the T cells, once they see the antigen, once they're presented with the antigen, and you can see kind of what's going on here. So let's say we have a macrophage, and it has eaten some kind of foreign cell, and some of those particles make it to the surface, right? And the T cell is then presented with that antigen and recognizes that antigen, right? And then you can see how that then stimulates the immune response. So in the case here, this helper T cell that's been activated then finds a B cell, and it activates the B cell to make more antibodies. So that's just one example. But you get the idea of antigen presentation is one cell presenting the foreign antigen to another immune cell so that that immune cell becomes activated and can then go out and do what it's designed to do. I like this picture over here just because it kind of shows you The red cell is supposed to be a lymphocyte, and the green cell is supposed to be an antigen presenting cell, like a macrophage maybe. And so they're showing you how that green cell kind of presents its antigens, the foreign antigens, to the red cell and says, hey, look what I found. Then that T cell becomes activated and can go out and do what it's supposed to do. What's important to understand as far as antigen presentation is that it's not just the antigen. So it's not just the foreign protein that's being presented. It's the foreign protein combined with a self protein, with a self surface protein. And we're going to talk about two of these self proteins, and they're both called major histocompatibility proteins. We don't like to say that word, and so we just say MHC, and we're going to talk about MHC1 and MHC2. So the MHC proteins are proteins that are normally on the surface of particular cells. But they are proteins that then kind of carry that abnormal protein, that foreign protein, and they present that protein to the lymphocytes. Now, MHC1, these are found on all nucleated cells. So that's most of the cells in your body, right? There's some cells like red blood cells, for example, that have no nuclei, and so they don't have MHC proteins. And that's why we didn't talk about MHC proteins when we talked about the antigens there. We talked about the ABO glycoproteins. And so here, for all the other cells, really minus red blood cells, you're going to have MHC1. So this is going to be really any self cell. So again, let's talk about you have a respiratory virus. You have a rhinovirus, which is a common virus that causes the common cold. So that virus is going to infect your respiratory epithelium, right? And your respiratory epithelia, they are nucleated cells. So guess what? They have MHC1 on their surface. And so when that virus enters your body, we're going to see this on the next page, enters that cell and tries to use that cellular mechanism to reproduce itself, some of its proteins get moved to the surface where they combine with the MHC protein, and then they can be presented to the immune system. MHC2 proteins are found, you can't even see that, can you? Basically, they're found on antigen presenting cells like macrophages, neutrophils, things like that. And we'll see here an example of each as we go through. Okay, so let's look at antigen presentation by MHC1 cells. So this is pretty much all of your regular cells. So let's get back to that respiratory virus. So let's say here is the respiratory virus. That's rhinovirus, let's say, and it's injecting its DNA into your body, and it's using the cellular mechanisms to reproduce some proteins that it's going to use to make more of itself. Eventually that makes its way through the endoplasmic reticulum, which is also, by the way, making MHC1 proteins. And those two proteins will combine, the MHC1 proteins. I skipped ahead, sorry about that. So you will get some of the viral proteins combining with the MHC proteins, and those will go to the surface. And now you have MHC1 proteins that happen to be holding out this foreign protein. So then a T cell that's floating by will recognize that complex and can then, if it's a cytotoxic T cell, it can actually destroy that cell. Plus the viral proteins, So this is a little bit different from antigen presenting cells. Remember, antigen presenting cells have MHC2. You can see MHC2 there. And so they're going to, take in a bacteria just like we talked about before. And that bacteria is going to be destroyed. And as it gets destroyed from the lysosome here, some of the kind of dissolved pieces will combine with MHC2 proteins that are being made, and then when they get to the surface, they've got a piece of that dissolved, let's say, bacteria. And now this antigen presenting cell can go to the lymph node, find a T cell, a helper T cell or something like that, and can present this foreign antigen to the helper T cell. So what we're going to find is that MHC1 proteins are really more involved with, let's say, viral infections, because it's the virus infecting your own host cell, and that causes a different kind of immune response. It causes that cytotoxic T cell immune response that kills your own cells in order to get rid of the virus. MHC2 is going to be more common, more present in bacterial infections, where those macrophages and neutrophils are out engulfing bacteria and then bringing those antigens to helper T cells, which can go to B cells, activate them to become plasma cells, and make antibodies specific against that bacteria. So MHC1 is more often seen in viral infections, MHC2 in bacterial infections. Here's just kind of an overview of what we've just been talking about, the MHC1 and MHC2. So remember, and I don't care that you know CD8 and CD4. I mean, you can remember those words, but I'm not really going to require that you have to know the differences there. So once a MHC1 type of cell that has a foreign antigen attached to it is discovered by a T cell, it depends a little bit on which T cell has come in contact with it. So if it's a cytotoxic T cell, it will actually attack and destroy the infected abnormal cell. If it's a memory T cell, it's actually going to go kind of into hibernation and wait to see that antigen a second time later on in your life sometime. And again, we're not really talking about regulatory T cells. If you have a antigen presenting cell with an MHC2 antigen complex, that's going to be presented typically to helper T cells, for example. Those helper T cells will then stimulate the immune response by cytotoxic T cells and B cells. Or you'll also get some memory helper cells that, just like the memory cytotoxic cells, will kind of wait for the next time that antigen enters the body. Okay, I wanted to talk a little bit just about this activation of B cells, because we kind of talked about the activation of T cells. So B cells, as I mentioned with a lot of the T cells, are typically in the lymph node, and they're in the lymph node closest to the site of infection. And so once they are presented with a foreign antigen, they become what we call sensitized. So that's kind of step one, is to be sensitized. Then once that sensitized B cell encounters a helper T cell, that helper T cell actually activates the sensitized B cell. So there's two kind of steps here. So antigen presenting cells are going to be the ones that sensitize. And then a sensitized B cell will encounter a helper T cell, and that will cause it to become activated. And once it's activated, it becomes a plasma cell and produces antibodies. Alternatively, it could become a memory B cell as well, right? So we saw with each of these cell lines, cytotoxic T cells, helper T cells, and B cells, that there are some memory cells that are created, and they're not really used for the current infection, but they're going to be useful for the secondary immune response. So here's a picture just kind of explaining that. So once the B cell encounters antigens, it becomes sensitized. Once that sensitized B cell encounters a helper T cell, it becomes activated. And once it's activated, it either becomes memory cells or it becomes plasma cells. And we see again that cytokines are involved in all of this as well. So here we can kind of see a interaction between the innate immune system and the adaptive immune system. Okay, so I keep mentioning this idea of primary versus secondary immune response, and I just wanted to go over it here with a couple of slides and talk about what are the differences. So the primary immune response is the first time. So if you've never seen that virus, you've never seen that bacteria before, this is the kind of response that you're going to get. And this response is both slower and weaker than a secondary response. And really, this is why we get sick often. The first time that we encounter a new virus or a new bacteria, often we get sick because our immune response is too slow and too poor, too weak really, to fight that infection in the first few days. So for the first few days, that bacteria or that virus is winning the battle because our immune response is too slow. Let's look at what happens, however, if you've seen this bacteria or virus before, and you have those helper cells, not the helper cells, sorry, you have those memory cells waiting. So you've got cytotoxic T memory cells, you've got helper T memory cells, you've got memory B cells, and they're all waiting to encounter that antigen for a second time. They're all on. Look here at how long it takes. So IgM we talked about was the first antibody that was produced, right? And it's taking, let's say that's 0 days, let's say this is 3 days. It's taking a few days to start, and it really doesn't peak until about 10 days. This is why we tell people, most viruses will last 10 to 14 days because we know it takes a while for that immune response to really get going. Look at the IgG. It takes about 5 days to start production, and it's almost 2 weeks before it's in full force. So really that 10 to 14 days is what we expect for a normal viral infection. But let's look at the second by. So what happens is you get a much quicker response and you get a much more powerful response. Look at what a huge response you get. First of all, you get a much greater magnitude response, right? You're still going to get some IgM and IgG, but look at how quickly they both start to be made on day 0. On the day that you encounter that antigen the second time, they begin responding. So you get a much quicker response because they're already there, they're already ready and activated. And so you're also going to notice, so you still get IgM as usual, but look at this huge IgG response. And remember, it's the IgG that's really doing most of the work in this adaptive immune response. And so the quicker and the greater amount of IgG antibodies that you can make, the stronger your immune response will be. And so often what happens when you're exposed the second time to the same virus, let's say, you actually don't get sick, right? You have what we say you're immune to that virus. And in fact, this is why we give immunizations, because we expose you to the antigen now so that if you see this virus 5 years from now, you will have a secondary immune response, which will be so fast and so fierce that you will likely not have any symptoms of infection because your immune system will attack that virus so quickly and so strongly that you'll never get sick from it. This is why we give immunizations. But this also happens naturally when you've seen, we talked about this in class, I think. If you had chickenpox as a child, you're very unlikely to get chickenpox as an adult because you have this secondary response on standby. This slide is just kind of an overview slide to give you an idea of the timing. So on the bottom axis here, we're looking at timing from 0 up to 3 weeks. So when you first encounter a foreign antigen, be it a bacteria, a virus, a fungus, a parasite, whatever, what happens first, what takes a little bit longer, and when do those activities peak? So you can see the first ones, we talked about this, especially for bacterial infection, right? For a bacterial infection, you're going to have neutrophils really being your first response. We didn't talk a lot about natural killer cells in the setting of a bacterial response, but they can be useful there. Macrophages, we did talk about. These are the phagocytes, right? Really, all of these are acting as phagocytes. So your phagocytes are really your first response. Once the macrophages have started eating the bacteria, they can then present those antigens to T cells. So now you start to get some T cell response, like cytotoxic T cells and helper T cells. And once those are activated, now your plasma cells become activated, and then you can start producing antibodies. So you can see there's kind of a timeline here, kind of a progression over time of which parts of this immune system are activated. And it's ultimately the antibodies that are going to kind of level most of your infections out, especially bacterial ones. So there's a couple of slides here talking about the difference between viral infections and bacterial infections, and we've talked about them a little bit, but I wanted to kind of review it here at the end. Oh, I think this, sorry, that's the slides coming up. This one's just talking about, again, an overview of cell-mediated versus antibody-mediated. It kind of gets us back to where we began. We said there were really two wings of the adaptive immune response. There was the cytotoxic side, and there was the antibody-mediated side, or cell-mediated and antibody-mediated. So when you get antigen presentation, that's going to activate T cells. Those T cells are then either going to become cytotoxic and actually directly kill the antigen cell, or it's going to form memory cells, and we didn't really talk about the T cells, the regulatory T cells. Or you'll get antibody-mediated. They're sensitized by the antigen, then they're activated by the helper T cells, and that produces plasma cells, which secretes antibodies, which also aid in destroying the antigen cells. And on both sides, you get memory B cells and you get memory T cells. So this is just kind of an overview of what we talked about. This is the slide I was talking about, giving you the difference between bacterial and viral responses. There's a lot of similarities here, so I just want to point out the differences. When you're talking about a bacteria, you're really talking about those first-line responders, macrophages, neutrophils, okay, antigen presenting cells. They're going to start the process by eating, right, phagocytizing those bacteria. Then they're going to present those antigens with MHC and they're going to present that to cytotoxic T cells, which will then come and destroy the bacteria directly. Or they can present to helper T cells, which then activate B cells, produce antibodies, and then we talked about the three functions of antibodies, right? They can either do opsonization and help with phagocyte activity, or they can form antigen antibody complexes and actually remove the bacteria out of the system. Virus is a little bit different because remember, viruses, the first step is infection of host cells. So you're not being, the antigen is not being presented by a macrophage or a neutrophil. The antigen is going to be found on the surface of a normal cell. So let's say you have an infected respiratory tissue, for example. There are some cytokines that are released, and that's going to have an effect. But what's going to happen is you're going to get appearance of those antigens in the plasma membrane, right? And two things can happen. Natural killer cells can see that foreign antigen and will just destroy the cell. We kind of talked about that in the innate system. Or activated cytotoxic T cells will see that foreign antigen on the normal cell surface, and it will also destroy. Now, there is some B cell activity going on and antibodies being made as well on the other side of this through the help of these helper T cells. So you can see there's some similarities, right? The activation of helper Ts and forming antibodies, that's pretty much the same on both sides. The main difference is MHC1 versus MHC2, and the antigens appearing on the surface of self cells here, and antigens here being presented to T cells on this cell. Okay, make sure if you have any questions, you can email them to me, or of course I'll see you in class on Monday and I can answer any questions you might have then. Otherwise, I'll see you guys on Monday. Make sure you do the influenza case as well as the study guide that I've put up for both the innate and the adaptive immune response.